Hello everyone. So today we are going to study about pharmacokinetic. It's a quantitative study of the movement of drug in the body. It includes absorption, distribution, metabolism and excretion. Absorption means how the drug enters our blood. Distribution means when the drug moves to the tissues and cells of the body. Metabolism is how the drug when enters the cell it is chemically modified, and excretion is how they are eliminated or removed from the body. The intensity of drug action depends on its concentration at the site of the action. Means how the drug will act, it will depend on how much it is present at the site of the action. Next we'll discuss about the biological membranes. They are made up of three things, that is phospholipid bilayers, the cholesterol and proteins and carbohydrates. So this is the phospholipid bilayer of the cell membrane. It contains cholesterol, phospholipids and various proteins and glycolipids. So it is divided into hydrophobic tail and hydrophilic head. Hydrophobic tail, it is inside, and hydrophobic head, it is towards outside. The function of the cell membrane is it acts as a barrier for the drug movement. Next is mechanism of transport of the drug across the cell membrane. How the drug is getting transport. There are two types, that is passive transport and the active transport. Passive transport are again of the two types, that is passive diffusion and filtration. The active transport are of two types, carrier mediated and vesicular transport. The passive transport itself says that it doesn't require any energy, but it will require a carrier. So no ATP is required, and it is the most common mechanism of drug absorption. While active transport, the ATP is required, and it will again also require a carrier for its transport. First we'll discuss what is passive diffusion. It is movement of drug from higher concentration to the lower concentration. So it doesn't require any energy, as I said, and it is not no carrier is required in this process, and it is not saturable. Means as no carrier is required, so it is not saturable. The factors which affect the diffusion process is its lipid solubility. Means the more the drug is lipid soluble, the more it will cross the membrane easily, because as the membrane is made up of lipid bilayer, that's why lipid soluble drugs are easily getting carried out through the membrane. Example is ethanol and thiopentone. Next we will discuss the next factor, that is the concentration gradient. Means if the gradient is greater, then the diffusion will be more. So greater will be the difference of the concentration, more will be the diffusion. Next we'll talk about how the pH and pKa will affect the diffusion process. So when the drug is unionized or non-polar drug, non-polar unionized drug means the drug is lipid soluble, more lipid soluble, hence it is easily absorbed through the cell membrane. But when the drug is ionized, means it gets polarized or ionized, then it becomes less lipid soluble, and hence it is not absorbed easily. Now let's discuss about what is Henderson-Hasselbalch equation. The formula is pH is equals to pKa plus log of A minus upon HA. What is A minus? That is the ionized form of a drug, and HA is an unionized form of a drug. What does this equation indicate? That it helps us to calculate how much of a drug is ionized or unionized at a given pH. At whatever the pH is given, whether the drug will be ionized or unionized at this pH. Now let's see what is the effect of pH on drug absorption. So weak acidic drugs like aspirin and phenobarbital, the absorption of these drugs occur better in acidic medium, that is the stomach, because the drug remains unionized in this medium. So acidic drug gets absorbed in acidic medium because it remains unionized in this medium. Likewise, when the weak basic drug is there, like atropine or ephedrine, it gets absorbed better in alkaline medium, that is the intestine. Now, what is ion trapping? So, when the drug enters the cell, when the drug it crosses the membrane and enters the cell, it becomes ionized inside the cell. So, the non-polar drug, when it crosses the lipid soluble membrane, it enters inside the cell, then it becomes ionized, and in this new environment it gets trapped. Example is aspirin, which is a weak acid. So hence it can't be removed outside. When it get ionized inside the cell, then it will not be removed outside, hence it may cause toxicity of that drug. Example is aspirin. It is unionized in stomach and it enters the mucosal cell, stomach mucosal cells. Aspirin is a weak acid, so it will absorb in the acidic medium, that is the stomach, and it goes inside the mucosal cell. Then inside the cell, because there is a slightly higher pH, it gets ionized there, and then it cannot diffuse back. This is known as ion trapping. Second, we'll study about filtration. It occurs through aqueous pores of the membrane. There are aqueous pores in the membrane, so filtration of a drug occurs through these pores. It depends upon the hydrostatic pressure, the molecular size. The higher the pressure, it will get easily filtered out, and also the molecular size. The smaller the molecular size, it will helps to easily filter out. No carrier is needed here also. This occurs in capillaries and glomerulus in the kidney. Now let's proceed further. We will discuss various carrier mediated transports. They are specific carriers are there for specific drugs. They are saturable, means they get saturated easily, and competitive inhibition is possible because for same carrier there can be 2 or 3 drugs going through the same carrier, so competitive inhibition is possible. First, we will discuss facilitated diffusion, as here no energy is required. Movement is along the concentration gradient and it is carrier mediated. So example is glucose transport by GLUT4 receptor. Second is active transport, which requires energy definitely, and the drug moves against the concentration gradient. Example is levodopa absorption and P-glycoprotein drug efflux. There are 3 types of active transport. First is primary active transport, second is secondary active transport, and third is vesicular transport. First, we will discuss the difference between primary active transport and secondary active transport. In primary active transport, the energy comes directly from the ATP hydrolysis. Directly ATP supplies the energy. And in secondary active transport, it comes from ion gradient, that is usually sodium ion, the gradient across the cell membrane, which gives energy to secondary active transport. Primary active transport example is ABC transporters, and the drug is P-glycoprotein. Function is to pump the drug out of the cell. If the drug is not required by the cell, so we can pump the drug out of the cell through ABC transporter. The location is intestine, liver, kidney, and blood-brain barrier. Now the secondary active transport are again of 2 types, that is the symport and the antiport. Example of symport is sodium glucose co-transporter, means the drugs are sent across the cell membrane. Both are sent across the cell membrane together. Antiport means they are sent in a different direction. Example is sodium calcium exchanger. Now what are vesicular transport? They are used for large molecules. This can be endocytosis or exocytosis. Endocytosis means taken inside the cell. So cell membrane engulf the substances forming a vesicle, and while exocytosis is vesicle is release the substances outside the cell. So example of endocytosis is vitamin B12 absorption, and example of exocytosis is hormone release and the neurotransmitters release. So that's it for today. In next video we will discuss the first parameter of pharmacokinetic, that is the absorption of the drug and how it happens. See you in the next video.